Why did shells and armor appear suddenly across dozens of lineages? In our last episode, you discovered how eyes evolved from simple light-detecting cells to sophisticated compound and camera eyes in less than 100 million years. Vision transformed ecosystems. Predators could hunt efficiently, and prey could see danger approaching. But seeing a predator isn't enough if you can't escape or defend yourself. This episode explores the defensive response to predation, the evolution of shells and armor. You'll learn how organisms incorporated minerals into their tissues, why hard parts appeared across multiple lineages simultaneously, and how mineralization became one of evolution's most successful technologies, persisting for 540 million years to the present day. Before the Cambrian, almost every organism was soft-bodied. Ediacaran creatures like Dickinsonia had no shells, no spines, no hard protective structures. They didn't need them. There were no predators. Then, within just 10 to 20 million years, hard parts exploded across the tree of life. Trilobites developed calcite exoskeletons. Brachiopods secreted hinged shells. Mollusks built calcium carbonate shells. Echinoderms formed calcite plates. Arthropods hardened their cuticles with minerals. Huliths, tomoshids, and dozens of other groups developed spines, cones, tubes, and scales. This wasn't random. This was evolution's response to a new, deadly challenge: predation. When Anomalocaris and other hunters began actively seeking prey, soft-bodied organisms became easy meals. The selection pressure was immense, and immediate evolved defenses or go extinct. Armor was the most effective solution, and dozens of lineages independently discovered how to incorporate minerals into their tissues. But building armor isn't simple. It requires extracting minerals from seawater, controlling crystallization at the cellular level, and balancing protection against mobility. Why did so many groups figure this out simultaneously? How do you build a shell? And what are the trade-offs that make armor sometimes advantageous, sometimes disadvantageous? The Cambrian arms race wasn't just about predators getting better at killing it, was about prey getting better at not being killed. And armor was the most visible, most successful strategy. Evolution from soft to hard. How did organisms learn to build shells and armor? The process required multiple innovations happening in sequence. The chemical challenge: building shells means incorporating minerals, usually calcium carbonate, charcoal, or calcium phosphate, into biological tissues. This sounds simple, but it's biochemically complex. Challenge 1: extracting minerals. Calcium exists in seawater, but it's dilute. Organisms need to concentrate it, pumping calcium ions from surrounding water into cells, sometimes against concentration gradients requiring energy. Early organisms probably developed calcium pumps for other purposes, regulating internal calcium concentrations for cellular signaling. Once these pumps existed, natural selection could repurpose them for shell building. Challenge 2: controlling crystallization. If you just add calcium to carbonate ions, you get random crystal formation, useless chunks without structure. Useful shells require controlled crystallization. Crystals must form in specific shapes and orientations. Growth must be directed to create desired forms. The structure must be strong, yet not too heavy. Organisms solved this using organic matrices, proteins that guide mineral deposition. These matrix proteins act as templates, causing crystals to form in precise patterns, layer by layer. The genes for matrix proteins probably evolved from existing structural proteins that happen to interact with calcium. Through mutation and selection, these proteins became specialized for shell construction. Challenge 3. Different minerals for different needs. Not all shells are the same material. Calcite, a crystalline form of calcium carbonate, hard, durable, relatively easy to precipitate. Used by trilobites, brachiopods, echinoderms. Aragonite, another crystalline form of calcium carbonate. Slightly less stable than calcite, but tougher, resists cracking, used by many mollusks. Calcium phosphate, used in vertebrate bones and some early shells. Different chemical properties, stronger in some ways, more metabolically costly to produce. Different lineages independently discovered different mineralization strategies, each with advantages and disadvantages. The timeline: rapid mineralization. Mineralized hard parts appeared rapidly across many lineages in the early Cambrian 530 to 520 million years ago. This timing correlates with increased predation pressure, as discussed in previous episodes. Rising calcium levels in seawater, ocean chemistry was changing, making calcium more available. Higher oxygen levels, more energy available for the metabolically expensive process of mineralization. These factors converged, and mineralization evolved independently in dozens of groups, almost simultaneously, a classic example of convergent evolution driven by strong selection pressure. Anatomy: diverse armor solutions. Let's examine how different groups built protective hard parts. Trilobite exoskeletons, calcite armor. We've discussed trilobites before, but let's focus on their armor construction. External structure: the trilobite exoskeleton consisted of calcite plates covering the dorsal upper surface. The calcite was arranged in 2 layers. Outer layer, dense, fine-grained calcite forming a hard protective surface. Inner layer, calcite with organic material interspersed, providing flexibility and toughness. This layered structure combined hardness, resisting penetration, with flexibility, resisting cracking. The exoskeleton was divided into articulated sections. Segments could move relative to each other, allowing the animal to curl, walk, or swim. The joints between segments were thinner, more flexible regions where the calcite was reduced or replaced by organic cuticle. Growth and molting. Trilobites grew by molting periodically, shedding the exoskeleton and forming a new, larger one. This process was dangerous. Pre-molt: the animal absorbed calcium from its old shell, weakening it. Molt: the shell split along predetermined lines and the animal pulled itself out. Post: for hours or days, the animal was vulnerable. Its new shell was soft and not fully mineralized. During this soft shell stage, trilobites were easy prey. Natural selection favored rapid mineralization. The faster the new shell hardened, the shorter the vulnerability window. Shell thickness variation. Trilobite shell thickness varied by species and environment. Thick shells in environments with intense predation, deep waters with eurypterids, areas with nautiloid predators. Thin shells in safer environments, oxygenated shallows with fewer predators, or very small species where weight was limiting. Thicker shells cost more energy and calcium to build and made the animal heavier and slower. Evolution balanced protection against mobility and metabolic cost. Mollusk shells, calcium carbonate fortresses, mollusks ancestors of modern snails, clams, and squids evolved shells independently from trilobites. External structure: mollusk shells consist of 3 layers. Outermost organic layer, protein-based, protecting the shell from dissolution and boring organisms. Prismatic layer, made of columnar calcite crystals arranged perpendicular to the shell surface, provides hardness. Nacreous layer, mother of pearl. Inner layer of aragonite platelets arranged in brick-like patterns provides toughness, resisting crack propagation. This composite structure is incredibly strong, much tougher than uniform calcite. The layered architecture stops cracks from spreading. Crack in the outer layer doesn't automatically propagate through the whole shell. Shell forms: mollusks evolved diverse shell shapes for different lifestyles. Univalves, snails, single spiral shell. The spiral shape is strong, structurally distributing force evenly. As the animal grows, it adds material to the shell opening, spiraling outward. Bivalves, clams, to hinged shells that can close tightly. When closed, the soft body is completely protected. Muscles hold the shells. Some species can resist enormous forces trying to pry them open. Cephalopods, early nautiloids, chambered shells with the animal living in the outermost chamber. Earlier chambers were gas-filled, providing buoyancy, allowing the heavy shell to float. This innovation made cephalopods successful swimmers despite shell weight. Growth: unlike arthropods, mollusks don't molt. They grow by adding material incrementally to shell edges, like adding rings to a tree. The shell grows continuously throughout life, never being shed. This continuous growth avoids the vulnerability of molting, but means the animal carries its entire shell. History injuries or growth disruptions are permanently recorded in shell structure. Brachiopod shells. Ancient bivalves. Brachiopods look superficially like clams' 2 hinged shells protecting a soft body. But they're not mollusks. They're a separate phylum that independently evolved bivalved shells. External structure: brachiopod shells consist of dorsal upper valve, usually smaller, ventral lower valve, usually larger, often with a whole pedicle forament where a stalk pedicle emerges, anchoring the animal to substrate. The shells are composed of calcite or calcium phosphate, organized in layered or fibrous structures. Internal structure: inside the shell, brachiopods have a lophophora, tentacle-like feeding structure that filters particles from water. The shell protects this delicate organ while allowing water to flow through when valves are slightly opened. Brachiopods were incredibly successful in the Paleozoic, more diverse than mollusks in many environments. Today, only about 400 species remain, but in the Cambrian and Ordovician, thousands of species existed. Spiny defenses: when armor isn't enough. Some organisms supplemented armor with spines extending hard structures outward to make themselves difficult to grab or swallow. Hallucigenia: we met this creature in episode 2. Its long, rigid spines running along its back made it nearly impossible for predators to bite or swallow without injury. Spiny trilobites: many species develop genal spines from the head. Thoracic spines from body segments, or pygidial spines from the tail. These made enrollment more effective, when curled spines projected outward in all directions. Echinoderm plates. Early echinoderms, relatives of modern starfish and sea urchins, developed calcite plates, embedded in skin, sometimes bearing spines. This created flexible, armor-protective, but not rigid like shells. Spines work through two mechanisms: physical deterrent. Handling a spiny organism injures predator metal parts. Size illusion. Spines make organisms appear larger than they are, potentially too big to swallow. Tube-dwelling, protective houses. Some organisms built mineralized tubes to live in, creating portable or fixed shelters. Huliths, extinct conical organisms with pyramid-shaped shells and an operculum lid that could close the opening. The animal could extend its feeding appendages when safe, then retreat and seal itself inside when threatened. Timoshiids, small cap-shaped or tube-shaped shells, often with elaborate surface ornaments. These are among the earliest mineralized fossils, appearing just before the Cambrian explosion fully kicked off. Tube-dwelling sacrificed mobility for protection. Once built, the tube couldn't be moved, but it provided near-complete safety when the animal was inside. Behavior. Living in armor. How did armor change behavior? Mobility costs, weight and drag. Armor is heavy. Calcite and aragonite are denser than seawater, so shells and exoskeletons create negative buoyancy. Animals sink unless actively swimming or have buoyancy compensation mechanisms. This changed behavior. Bottom dwelling became common. Many armored organisms adopted benthic bottom-dwelling lifestyles. Crawling on the sea floor doesn't require fighting gravity already down there. Swimming became specialized. Armored swimmers needed powerful muscles to overcome shell weight. Nautiloids solved this with gas-filled chambers. Some trilobites developed flattened hydrodynamic shapes, reducing drag. Burrowing intensified. If you can't swim well and crawling is risky, go underground. Many armored organisms became burrowers, hiding in sediment where predators couldn't reach them. Feeding limitations. Can't eat and hide simultaneously. Organisms in shells face a dilemma. To feed, they must expose vulnerable soft tissues, tentacles, siphons, mouth. But exposure means vulnerability to predation. Solutions. Filter feeding from within shells. Bivalves and brachiopods pump water through slightly open shells, filtering it internally. This minimizes exposure time. Rapid retraction. When threatened, soft tissues pull back inside shells instantly. Muscles hold shells closed until danger passes. Nocturnal feeding. Some organisms fed at night when visual predators were less active, remaining sealed during the day. Armor enhanced boldness. Organisms with excellent armor could be more adventurous, grazing openly, crawling in exposed areas because they had protection if attacked. Reproduction. External fertilization still dominant. Armor didn't fundamentally change reproduction. Most organisms still released gametes into water for external fertilization, but shells required modification. Spawn through openings. Shell openings allowed gamete release during breeding seasons without compromising protection. Synchronized spawning. Releasing eggs and sperm simultaneously increased fertilization. Success important when gametes must travel through water to meet. Armored organisms couldn't develop elaborate mating behaviors, requiring direct contact. Shells got in the way. This limitation persisted until some lineages, certain mollusks, arthropods, developed ways to transfer sperm directly. Social behavior, minimal armor didn't promote social behavior. Shelled organisms remained largely solitary, occasionally aggregating in favorable locations, but not cooperating or communicating in sophisticated ways. Why? Because armor reduced the need for social defensive strategies. Animals without armor, like fish, later benefited from schooling many eyes, detecting danger, confusing predators through numbers. But armored animals could defend individually, less pressure for social evolution. Adaptation, armor for every need. Armor adapted to diverse environments and threats. Adaptation to predator type, crushing versus piercing, against crushing predators, jaws, claws, thicker shells, reinforced ribbing, adding structural strength, rounded shapes distributing force evenly, against piercing predators, drills, spines, layered construction, a piercing tool breaking, the outer layer doesn't automatically penetrate inner layers. Regular shell repair organisms continuously thickened shells from inside, sealing weakened areas, against chemical attack boring organisms, acidic secretions, organic periostracum coating protecting calcite from dissolution. Rapid shell repair, constantly adding new material to maintain thickness. Adaptation to environment, water, chemistry, and depth. Well calcified waters, tropical, shallow seas. Thick shells, calcium is abundant. Shell construction is cheap. Elaborate ornamentation, spines, ridges, colors, poorly calcified waters, deep, cold, or acidic. Thinner shells, calcium is limited. Thick shells, too costly. Simpler forms, less ornamentation. Some organisms abandon shells entirely, returning to soft-bodied forms. Deep water, thinner shells, less predation pressure at depth, less mineralization, saving energy in low food environments. Adaptation to lifestyle. Mobile versus sessile. Mobile organisms crawling or swimming. Lighter shells minimizing weight. Streamlined shapes reducing drag. Flexible joints allowing movement. Sessile organisms attached to substrate. Heavier shells, weight isn't a disadvantage when you're not moving. Irregular shapes fitting onto rocks or other attachment surfaces. No need for symmetry, function matters more than form. Adaptation over time. Escalating arms race. As predators evolved better weapons, prey armor had to improve. Early Cambrian, simple thin shells, basic spines, sufficient against early predators with weak jaws. Middle Cambrian, thicker shells, more elaborate spines, responding to predators like Anomalocaris with powerful grasping appendages. Late Cambrian, Ordovician, very thick shells, complex hinges that lock shut, chemical defenses supplementing armor, responding to nautiloids with crushing beaks and early jawed fish. This escalation continues today. Modern shell-crushing crabs faced mollusks with incredibly thick shells, and both are more heavily armed than their Paleozoic ancestors. Extinction. Why some armor types disappeared. Not all armor types persisted. Some disappeared. Why? The calcite crisis. Permian-Triassic extinction. The Permian-Triassic extinction 252 million years ago killed over 90% of marine species. Among the hardest hit were calcite-shelled organisms. Why? Ocean acidification. Massive volcanic eruptions released enormous CO, acidifying oceans. Acidic water dissolves calcite, making shell construction difficult or impossible. Organisms with calcium phosphate structures or organic armor survived better. Calcite-dependent groups suffered mass extinctions. This pattern repeats at other extinction events. Ocean acidification disproportionately affects calcite-shelled organisms. The decline of brachiopods dominated Paleozoic seas, outnumbering bivalve mollusks. But after the Permian extinction, bivalves diversified while brachiopods declined. Why did bivalves outcompete? Bivalves had advantages. Active burrowing brachiopods, mostly sessile, muscular foot for movement. Siphons allowing deep burial while still feeding, more efficient filtering systems. Greater anatomical flexibility allowing diverse lifestyles. Brachiopods, once dominant, are now ecological relics, still existing, but relegated to marginal environments where bivalves are rare. The persistence of mollusk shells. Mollusk shells evolved in the Cambrian and remain standard today, 540 million years of continuous use. Why such longevity? Because the mollusk shell design is near optimal. Layered structure providing strength and toughness. Continuous growth without molting, avoiding vulnerability. Incredible diversity of forms: coiled, bivalved, conical, chambered. Compatibility with diverse lifestyles: mobile, sessile, swimming, burrowing. Mollusk shells are evolutionary perfection, not unchanging, but so fundamentally sound that variations on the theme remain competitive even after 500 million years. Current status: armor still dominant. Mineralized shells and armor remain standard defensive strategies today. Mollusk shells protect snails, clams, oysters, arthropod exoskeletons, armor insects, crustaceans, arachnids, echinoderm plates protect sea urchins, starfish, vertebrate bones, mineralized internal skeleton support, fish, reptiles, mammals. The Cambrian innovation of biomineralization never became obsolete. It remains one of evolution's most successful technologies. Shells and armor transformed the Cambrian seas from a garden into a fortress. But armor created new niches. Borers evolved to drill through shells. Crushers developed jaws to break them, and some predators learned to inject toxins, bypassing armor entirely. In our next episode, we'll explore the Burgess Shale, one of the most important fossil sites in the world, preserving exceptional detail of Cambrian life, including soft-bodied organisms rarely fossilized elsewhere. You'll see creatures so bizarre they defy easy classification, and you'll understand why the Cambrian was evolution's most creative period. The arms race continues. The fossil record reveals its history, and some fossils preserve that history with breathtaking clarity. This is HistoPrimordia, where history comes alive. Thanks for watching. Subscribe now and join us for the next episode's story.